With that said, again, our first part of the symposium will be on organ development. And our first speaker is well known to most, if not all of you, and it's Dr. David Cornfield, who is a world-renowned developmental lung biologist slash clinician-scientist. He is a professor, obviously, in pediatrics, and actually his research program is focused on basic and translational and clinical research with a focus on lung biology. He's mentored countless trainees who are now division leaders around the country and around the world. He's a leading expert in thinking about how the lung is developing both in utero and in children. And obviously this has tremendous ramifications, not just for lung disorders in utero, but really a whole slew of other diseases, including prematurity. And so we're really, really excited to have David here today. And with that, David, the floor is yours, and you got about 15 minutes. Thank you.
Great. Well, thank you, Yasser, for that very gracious introduction. And I applaud the whole program that everyone's undertaking in order to really create something that's greater than the sum of the parts. Hopefully you can see my slides. Is that all good? So what I'd like to do today is I'd like to focus on something that is perhaps both the past and the future. So I'd like to speak about an area that I've been focusing on really for the last too many decades to admit, and that's really the pulmonary circulation in transition. And I'd like to really go at this from the past, where we're going to talk about the whole organ and whole organ physiology, to the molecule. And in the hopes of introducing these concepts that perhaps we can accomplish some of the goals that Yair set before us, which is to create collaborations. So this is a slide that really is a very famous slide of the transition of the pulmonary circulation. It's perhaps the first data that really underscored this whole idea that, and this was from Sir Geoffrey Dawes, as you can see, it's from a J. Physial paper from 1953. And in this, he used a fetal lamb that he delivered, and he demonstrated that with the onset of ventilation, there was a substantial increase in pulmonary blood flow, and there was a dramatic decrease in resistance. Everyone who's been in the delivery room, either as a patient or with a mom, a dad, or as a doctor, recognizes this is one of the most remarkable moments of life. The fetus moves from this low oxygen tension environment to one of a much higher oxygen tension environment. The pulmonary vascular resistance is very high, and pulmonary blood flow in the fetus is less than 10% of the biventricular output. And within the first few moments after birth, there is this remarkable transition that ensues. Oxygen tension increases by about 5-fold. The pulmonary blood flow increases by about 8 to 10-fold. Pulmonary artery pressure decreases by 50% in the first 24 hours of life. And this is a remarkable transition that is virtually unprecedented at any time during the organismal survival. And it's a transition in the pulmonary circulation that never is repeated again for all of our air-breathing lives. And, And how this transition unfolds has been the subject of intense investigation for many, many years. And it's really the story, in some ways, of one of, in my opinion, one of the most important translational biology, translational therapies that we've really been able to bring to the table in the last 30 years. And I want to tell you a little bit about that story, and then talk a little bit about how we got there, and also then talk a little bit about where we might be going in the future when we really are focusing on the transition of pulmonary circulation. So as I talked about, in utero, the pulmonary artery really doesn't get much blood flow. The endothelial cells are bunched up and they don't allow that much blood flow. But at birth, with the onset of air-breathing life and oxygenation, shear stress, ventilation, and the establishment of an air-liquid interface, the transition unfolds. and there's a dramatic decrease in pulmonary vascular resistance. We, many years ago, demonstrated in our version of fetal surgery, which we did in fetal lambs, that if you go ahead and you deliver a fetal lamb and you give ventilation, there's a dramatic increase in pulmonary blood flow. There's a significant decrease in pulmonary artery pressure. And if you block nitric oxide and prevent its elaboration, then that increase in pulmonary blood flow didn't ensue, and pulmonary artery pressure also didn't go down. We were also able to show in a model of chronically instrumented fetal lambs that if you compress the ductus arteriosus and increase mean pulmonary artery pressure, there's normally this dramatic increase in pulmonary blood flow. Shear stress-induced pulmonary vasodilation. Blocking nitric oxide prevented that. And in many ways, this was sort of the background for the studies that we were doing where we're giving nitric oxide to infants. And it was, for those of us who can remember who were in the delivery room or neonatologists among us, a dramatic story of success. But really, we didn't know exactly how nitric oxide really worked. We had some thoughts about it, and we really didn't know why the pulmonary circulation was able to sense and respond to oxygen in such a dramatic fashion. And so for the next many years, my lab embarked on trying to figure that out. Among the things that we found, this is cytosolic calcium, which is a metric for the contractile state of pulmonary artery smooth muscle cells, that fetal, as opposed to adult pulmonary artery smooth muscle cells, responded to an acute increase in oxygen tension with a significant decrease in cytosolic calcium, which would correlate with a decrease in the contractile state. We were also able to identify, using electrophysiologic studies, that there was a spontaneous transient outward current that existed only in fetal and newborn pulmonary smooth muscle cells and did not exist in the adult pulmonary smooth muscle cells. Further, we used confocal microscopy to determine that there were these presence of something called sparks, these very finely tuned aliquots of calcium that were released from ryanodine-sensitive stores in the pulmonary artery smooth muscle cells to cause activation of a potassium channel and therefore pulmonary vasodilation. And we elaborated this story over the course of many years, that either nitric oxide or oxygen would cause a subcellular signaling cascade that resulted in release of a very localized spark of calcium immediately adjacent to this calcium-sensitive potassium channel, which would lead to the elaboration of potassium membrane hyperpolarization and vasodilation. And that was a very satisfying set of physiologic and molecular experiments. We also wanted to kind of determine, well, how does the fetal lung and the pulmonary vasculature, the mesenchyme, actually prepare to have this remarkable sensing and oxygen tension that allows for air-breathing life to exist? So we've now, for the last several years, embarked on a project to test the hypothesis that these lung mesenchymal cells have a dramatic increase in diversity after birth, and that there's a change in the molecular signature that really determines cell function. So to test this hypothesis, we went ahead and used a relatively new strategy, though it's certainly not that new at Stanford, and that is single-cell Rna-Seq. We went ahead and used mice from different different ages, from late fetal. A mouse delivers at 19 days or so right before birth, at 1 day of life, at 7 days of life, and at 21 days of life. We were able to perfuse the lungs. We dissociated the cells using FACS. We went ahead and generated libraries, and then we used NovaSeq to make reads, and we mapped the reads to the genome, and we were able to figure out exactly what genes were present in what transcriptome was present in which cells. And these are the people that did all of that work. And we also have undertaken in situ validation of these findings. So first, this is not our work. This is a correlation of many people's work, but this just really shows you that the lung possesses rather tremendous cellular diversity. And this UMAP on the left hand of your slide really is colored such that you can see the relative abundance of these different cells. We also found that the lung mesenchymal cell diversity increases dramatically after birth. These are the cells that are present in the lung mesenchyme at E18.5, just these three cellular subtypes. And within the first day of life, there is this dramatic increase in the number of cells, and this increases progressively over the first 21 days of life. It continues such that we have this development where you can see there's the perivasculature and there's early and late fibroblasts, and there's multiple cells that comprise the lung that are really increasing in both diversity and complexity over the first 21 days of life, such that they really inform the alveolarization of the lung. This lung mesenchymal cell population is very dynamic, and it's specifically dynamic during this early stage of life, but especially during the stage of alveolarization. In the mouse, P7 really correlates to probably about gestational age 32, 33 weeks in the human at the onset of alveolarization. And so this really permits us the opportunity to study the alveolarization that's occurring in human infants using the mouse model in an air-breathing life sort of fashion. This maturation occurs in a sequential fashion, and it moves outward. You can see these are the only cells present in fetal life, and there's an increased complexity over the first 21 days. We also were able to look at the transcriptome, and this is these plots demonstrate that specific oxygen sensing genes change rather dramatically. This is the gene that encodes the protein for hypoxia-inducible factor 1, the transcription factor, hypoxia-inducible factor 1 alpha, and actually hypoxia-inducible factor 2 alpha. And you can see that there's this huge increase. Lighter is hotter, and this is going from E18.5 to P1. And this is for the endothelin receptor B, another very important gene relative to regulation of pulmonary vascular tone, as is the case with myosin light chain kinase, and then PDE5, multiple genes that play a critically important role in the regulation of vascular tone, and also with respect to vascular remodeling as well. These cells are specifically enriched for genes that seem to play an important role in their function. Early myofibroblasts, and you can see them denoted here, are enriched for genes that really play a role in cell commitment and alveolarization, and loss of these genes, which may be happening when people give dexamethasone, may compromise alveolarization. There's also a proangiogenic component to these cells as well. These cells, these are the myofibroblasts, are spatially distributed in a manner that seems to be important for determining remodeling of the lung and actual alveolarization, or the secondary septation that occurs. And the later cells are adapted really to allow for remodeling of the lung. And you can see that here are the early cells that are more embryonic, and they have these smooth muscle-like qualities. And later on, they become anti-angiogenic as there's not as much need for angiogenesis to occur. And you can see that still later on in the more P21 stages, there's some collagen synthesis genes that are present. In a finding that we think is quite remarkable, we were able to identify a single cell population that elaborates corticotropin releasing hormone. This is a previously unreported finding. And our postulate is that corticotropin releasing hormone, you can see it's demarcated right here, is present only at E18.5. We believe that it is sending a signal into the bloodstream to the brain to cause release of ACTH, which then allows for glucocorticoid release. And that is a really important signal, as everyone I think knows, for surfactant maturation. And this really is the first evidence, if we obviously have to publish it, that the lung itself is almost capable of maturing itself. So I think I was good to the time, I hope, here. And I just like to conclude in saying that the lung mesenchyme has remarkable heterogeneity. There's transcriptomic alterations that are temporal specific, and they have a lot of physiologic and I think therapeutic implications. There's a fibroblast molecular signature that changes during development and has a spatial distribution. And this molecular signature informs cell specialization, function, and location. And whether the dynamic transcriptomes of the lungs are pre-programmed or respond to physiologic stimuli remain unknown. And I'd like to just highlight my collaborator and partner in all this, Christina Alvira, who's done so much of the heavy lifting. I'd also like to acknowledge another partner and collaborator, Steve Quake, who's just been really wonderful, and Fabio Zanini, one of his postdocs, who's now one of our collaborators, and he's at the University of New South Wales. And then finally, Shibing Che, one of the scientists in our lab, who's really done an incredible amount of this work. So thank you, and I'll stop my share at that point.
Thank you so much, David. I'm sure so many people may, I certainly have my own questions. I'm sure so many people have a lot of questions, but again, please save those questions for the end of this section. We have two more speakers, and then we should be able to get to some Q&A at around 6:15 or so, maybe even earlier. But thank you so much, David. This was fantastic. That's exactly what we were hoping for. Our next speaker is—
Thank you for the invitation.
Our pleasure. Our next speaker is Dr. Carl Sylvester, who, from a clinical perspective, is my pediatric surgery colleague and right-hand person who's just a phenomenal person to work with and collaborate with, and I just want to acknowledge that. And obviously he's a superb clinician and basic science and translational science investigator, and that's what he's going to talk to us about today. He's a professor in the Department of Surgery here at Stanford, and his interest is primarily on human development, and specifically recently on metabolomics, and he helps to lead that area of inquiry across multiple disciplines. He is interested in GI development as it relates to this particular area and many other areas, but he'll talk to us about that today. So if we think about organ function, we just heard about the lung, and the gut, of course, is just as important, and we have many anomalies and other conditions that we collaborate on, and we're looking forward to his talk. So thank you so much, Carl, and I'll mute myself now.
Thanks, Jair, for the invitation, and thank you also to the Dunlevie leadership and organizers for inviting me to present tonight. A lot of familiar faces in the group. I took it as my assignment to talk a little bit about the gut, among other things, but I'm also going to coin a term tonight, which is going to be newborn gut dysfunction, which is unfortunately the state of affairs today in many of our newborns, whether they're hospitalized or not. So to get us rolling, I'd like to give a nod to actually current events, which is the COP26 occurring, the UN Climate Change Conference in the UK currently, where world leaders are discussing climate change, global ecologic extinction and loss of biodiversity. And here at home and in the healthcare industry, these same industrial innovations actually have put pressures on our gut microbial diversity, which is depicted in this black line as we become more and more urbanized in society. And as a consequence, there's been a lot of associations drawn to the increase in modern diseases, which have now shifted away from acquired infectious diseases to acquired chronic autoimmune and inflammatory diseases. And if we bring it closer to home and we say, just on the left-hand side of this slide, what's going on with our infants, we've identified a profound change in breastfed infant gut microbiome due to a reduction of bifidobacterium over the past century. And at first pause, why is that a big deal? You might question. Well, this little graphic here demonstrates actually some of the outward measures and pressures that have been placed and the result of that. And very simply, the introduction of formula feedings, antibiotic usage perinatally to both mom and baby, as well as C-section use, has resulted in a dramatic disruption of the transfer of health-promoting microbes from mom to baby, and the loss or lack, although reintroduced now, of formula feeding for breast milk has resulted in further deselection pressures. And the ecological changes, simply measured by a pH change, where historically and physiologically, the fecal pH should be around 5 to 5.5, is now measurable in the 6 to 6.5. And that's as a result of some significant change in the short-chain fatty acids that are being produced in the gut. So the depiction here on the right is that when you've got a lack of very health-promoting bifidobacterium, as addressed by the panel on the left, you get an overabundance and expression of Enterobacteriaceae. And this is the taxa of bacteria that includes all those bugs that we typically consider potential pathogens, like E. coli, Klebsiella, and various enterobacters. And so again, the last ecological metaphor here is that this niche is matching these species to a specific environment, in this case, the gut of the newborn, relative to the resources that are available to them and how they might outcompete their competitors. And unfortunately, we're really in the red at this case, as opposed to in the blue, which is really what nature intended. And so why do I say that? Well, if we look at breast milk for a moment, you can see here on the left is that the vast majority of it is water, but of the macro and micronutrients, the vast majority of that, which is lactose, lipids, and protein, making up 85% of the dry weight, is food for baby. That's very clear. What's little less understood is actually this other component known as HMOs, or human milk oligosaccharides, is actually food for the bugs, food for your gut microbiota. And if you think about it, that breast milk was really the only food source available to human beings for tens of thousands, if not hundreds of thousands of years of evolution. And so why would mom expend so much energy to create this 15% of these macronutrients that are specifically for bacterium? So precisely, humans have no ability to digest or metabolize HMOs. Rather, bifidobacterial species carry all the enzymes to break down these 5 monosaccharides, which are depicted here on the right, into their building blocks and provide them as both a fuel source as well as additional metabolites that may provide some benefit to the baby. And that's what we're going to spend a little bit of time talking about. And so we published a paper earlier this year, which was a survey across the United States. We collected poop in pediatrician's office on babies who were not exposed to antibiotics. And we broke it down by regions. And what we identified is that, in fact, there was a significant lack or loss of bifidobacterium, and there was a favor or favoring or a shift towards Enterobacteriaceae depicted in the red, and the bifidobacterium, to keep it simpler, depicted in the blue. And no matter where you looked across the United States regionally, this was pretty much the same pattern. And it was also independent, for the most part, of whether you were exclusively breastfed, you had formula feeds, or you were mixed feedings. And when you summed it all up, on average, bifidobacteriaceae was present in only about 19% or 20% of relative abundance across these babies. And potential pathogens like E. coli and Klebsiella in this taxa of Enterobacteriaceae made up the vast majority of the others. So why is that problematic? Well, that's true not only, and these are term babies who are outpatients, it's also true of hospitalized babies. The taxonomic tree on the right is our hospitalized babies. Almost no matter where you look and what NICU in the developed world, you'll see a shift on this tree from what should be there, from firmicutes and actinobacteria, where the bifids and the lactobacilli live, to the right side, which is proteobacter. And again, that's all the Enterobacteriaceae and the potential pathogens. And some very elegant work that was done by Barb Warner and Phil Tarr out of WashU St. Louis is followed up by Joe New at the University of Florida, in fact, demonstrated that this bloom, or this preponderance of proteobacter, was always there and present prior to the onset of necrotizing enterocolitis. And those of you who have seen me speak before on anything research know I can't give a talk without speaking at least for a moment about necrotizing enterocolitis. Yet this association really doesn't make any sense if all the babies are heavily colonized by proteobacter because of the pressures of antibiotics, largely because bifids are very, very sensitive to them. And as soon as you withdraw the antibiotic, all the proteobacter come back in and the bifids have no chance. So why is it that some babies get necrotizing enterocolitis and some babies don't? So to address this, rather than just looking at what the bugs that were there, we asked the question of maybe it's what they're doing. And specifically, bacterial fermentation of these feedings are altered in the newborn, and that's what puts them at risk for NEC. And more specifically, this intersection of being premature, which means your gut's not developed, you have enteral feedings coming in, and there's sometimes maybe perhaps held up where NEC occurs in the terminal ileum because of the ileocecal valve, and you get microbes there. So you set up this fermentation reaction. So to gain evidence for this potential hypothesis and explore it, to summarize a tremendous amount of work over several years, is we gathered the publicly available databases for metagenomic sequencing in babies who were gestationally age matched for either necrotizing enterocolitis and control. And in work done here at Stanford, we collected the poop on babies serially, those with necrotizing enterocolitis and those without, as well as those babies who had recovered. We looked at the metabolite composition of their fecal streams, and then we used those fecal extracts and specific metabolites that we identified to do preclinical testing in animal models and humanized cellular models. And what we found is that indeed, number one here was that newborn gut dysbiosis, typically defined by a lack of diversity and a concentration of these Enterobacteriaceae species, was true. But the new findings were that it was actually this enrichment in specific metabolism pathways, and particularly this pyruvate formate lyase was heavily enriched significantly in those babies who developed necrotizing enterocolitis. And the result of this is that instead of pyruvate moving through the various glycolytic and transfer chains for energy development, PFL actually metabolizes pyruvate to formate, which is a bacterial mixed acid fermentation producing a lot of carbon dioxide and methane, which are precisely the gases that we see in the baby's X-ray who has pneumatosis and necrotizing enterocolitis. And moreover, in data that I won't show tonight, when we expose these various preclinical models, both in a mouse model as well as humanized cellular organoids, we found that the mechanism of action of formate is the reduced mitochondrial ATP production, increased epithelial monolayer permeability, and produce a histologic picture that very much looked like necrotizing enterocolitis via cell death pathway known as necroptosis. So what can we do about this? If all our babies are heavily colonized by Proteobacter, and I've told you that nature has actually created a system where these bifidobacterium should be there to take these HMOs and to convert them to short chain fatty acids, we look at this particular bug known as B. longum infantis. And the mechanism here is that it has the machinery or genetic capability to capture HMOs extracellularly, which are flowing through the gut of the baby who is being fed, transport them intracellularly to remove them from the competition of other bugs that are present, and fully digest them to lactate and acetate, which accounts for that very low pH that I spoke to in the earlier slides. So absent this mechanism, everything shifts to a high pH favoring Enterobacteriaceae, and the bifidobacterium are not there. As long as we have HMO and this bug is introduced, particularly those that have an H5 positive gene cluster for the transport and the full utilization, this bug is able to outcompete all the other competitors as depicted here on the right. In this case, it's a competitive assay versus an H5 negative bifidobacterium. So even within the same taxa, within the same species and strains, we see this difference based on the genetic endowment for this ability to consume HMOs, a key ecological feature when we're competing for gut colonization and overall health promoting effects in the newborn. So what this slide shows is that you can measure these effects. In any baby who receives bifidobacterium supplementation, you can measure that if they don't receive it, as depicted in the gray bar, all the HMOs are washed through into the stool. And this can create things like, we think, colic, diaper rash because of the shifts in pH, and a variety of other things like necrotizing enterocolitis if you happen to be a very premature and vulnerable baby. In contradistinction to that, if you have bifid colonization and being supplemented, you see an increase in the total short chain fatty acids, largely lactate and acetate. And by the same token, you see that when you've got extensive colonization by Bifidobacteriaceae, they outcompete and they displace all the potential pathogens of Enterobacteriaceae. We also know that if you are heavily colonized by Proteobacter and Bacteroides, these guys actually favor digestion of the mucin glycans, which makes up your mucus layer of your gut, unlike the taxa of B. infantis, who eat the HMOs, preserving the mucus layer here and allowing for a much more intact epithelial barrier. This again can be measured looking at the fecal stream, where you can see in those bugs, those babies who are not supplemented, they have a lot of digestive products of the mucin glycans as depicted in the gray bar here. Whereas if you are supplemented, you produce a lot of acetate and lactate as depicted here on the left. You reduce the amount of formate that's being produced as just an example of the four short chain fatty acids that predominate in these various reactions. So we've set up a series of experimental or empiric tests where you can take the supernates or you can take the fecal waters that are pooled from babies that are colonized by Enterobacteriaceae predominant versus those that are colonized by Bifidobacterium predominant. These are sterilized and they are then strained and they're placed in these various preclinical enterocyte models. And whether it's a cell line that's immortalized, a primary cell line, or human intestinal epithelial stem cells, what we find is that these supernates actually provide for a survival advantage when the cells are challenged by something like LPS, or in the case of the far right here, when we have formate added at certain physiologic concentrations that we're able to measure in the gut of the infant, we see that those babies who are at least exposed to the effluent of what these bugs are pumping out in reaction with HMO has a protective effect. Using the same type of setup, what we also know is that this favors cellular respiration, largely due to the acetate and lactate that is being produced in abundance and the diminution of formate, which uncouples the electron transport chain. We see that at basal respiratory status, you see that EBC001 fed babies, because of the supernate, have a much greater production of ATP as reflected by oxygen consumption rate. If you add oligomycin to unlink this, you see that actually the vast majority of what we're measuring here is as a result of mitochondrial respiration and ATP production. And in fact, it better prepares the cells for a stimulus that says, you know what, there's something going on here, we need more energy. And this is the max respiration response that was all measured on the seahorse respirator. So in essence, what we think is going on here is that these bacterium interacting with HMO, not only favoring the colonization, reducing the pH, but also producing these various metabolites and mediators, which is going to favor the intact preservation and the physiologic function of the epithelial lining themselves. Now, work that is ongoing and exceeds what we have time to speak to tonight is this paper that came out in Cell earlier this year, work by our collaborators at the Karolinska, as well as at Evolve Biosciences, demonstrated that in those babies with this graphical abstract that had high HMO utilization, there was a significant shift in not only innate, but also adaptive immunity, specifically through the byproducts of HMO utilization by these bifids. And they narrowed it down to a few candidates, including indole-3 lactic acid, which is a metabolite of tryptophan, and galectin-1. And what they found was that when you had high HMO utilization in byproducts versus low, you ended up with a very Th1 or T cell subsets that was associated with low inflammation and immune tolerance. Conversely, if you had low bifidobacterium and you had low HMO utilization, you had T cell polarization times the T helper 17 and 2 subsets, which favors autoimmune and allergic diseases. Other groups have extended these findings further in work showing that indeed bifidobacterium presence in the breastfed milk baby versus the Enterobacteriaceae or Bacteroides in this case is inversely associated with your prevalence or predisposition to autoimmune and allergic diseases. So to summarize a tremendous number of different strings that I'm pulling on here is that infant gut health and this relationship between mom and baby begins with breast milk and goes far beyond the nutrition for baby, when in fact it actually has this HMO which selects for these health promoting bacteria known as bifidobacterium. They consume the HMO, they displace the proteobacter, decrease the inflammation, promote the mucus layer, protect the epithelial barrier, and alter systemic, potentially immune programming that would then affect the onset of disease. The specific example I gave you in the premature babies on the left-hand side of this spectrum down here was necrotizing rhinocolitis. So ongoing and future forward-looking work is going to continue to unravel the metabolic interface here and the impacts on the immune system for potentially proper immune development and things when we think about life course, the effects on an atopic march most specifically, starting with asthma, moving on to things like eczema or type 1 diabetes and Crohn's colitis over a much larger arc of the healthcare. Importantly, all of this is taking place consistent with the Dunlevie theme at birth. And the window to be able to impact this begins with those very first feedings in inoculating baby with the potentially health-promoting bacterium and allowing for this natural symbiont to develop, displace those pathogens, and then program the immune system as well as providing the gut with a protective barrier. So with that, I'll wrap. I just want to give a shout out to the Metabolic Health Center research team who's been contributing dramatically to these studies, which we're looking to take forward with collaborators not only in MFM, but also in neonatology. Folks from my lab, as well as collaborators from Evolve Biosystems, Bethany Henrik and Steve Fries. And finally, the website for the Metabolic Health Center, where we'll be continuing to move into studies together with our collaborators in OBGYN, maternal fetal medicine, looking at the effects of various maternal covariates on the same types of relationships. Thank you.
Thank you, Carl, for this outstanding and very comprehensive and tightly packed overview of so many aspects of gut health. You took this in a completely different direction than I thought you would, and opened up so many different areas of questioning. But we'll save it until after one more speaker for this particular session. So again, we heard about the lung, we heard about the GI tract, and now we're gonna hear about the developing brain. And Anca Pasca is a superb neonatologist. She's an assistant professor in the Department of Pediatrics and Neonatology and Developmental Medicine. And she's becoming a very, very prominent sort of neurodevelopmental biologist, if you will, studying the molecular mechanisms of neurodevelopment as they relate to a variety of conditions, pathological conditions, both in utero and genomic and otherwise. And we're really excited to have her here. So without further ado, Anca, I'll let you take it from here, and then we'll do some quick Q&A with you and the other speakers. So thank you so much.
Thank you very much for that very kind introduction. And I'm very honored to be among the speakers, and I'm really excited about this initiative. It's really, really exciting. I hope you can all see my screen. And as Yair said, I'm going to talk about human cellular models for brain development, and specifically for brain injury of prematurity, which has been my primary area of focus so far in my just newly established lab. And I'll start by introducing you to Haven and his mother, Amanda. Some of you might know this baby. He was delivered at Packard. He was very small for gestational age, delivered extremely preterm at 25 weeks, and he spent about 5 or 6 months in the NICU. A lot of you might have been actually helping him survive during this period of time. And as a lot of our extreme preemies, he went through a lot during his NICU stay. He had respiratory failure. He had a lot of hemodynamic instability, intestinal surgeries for multiple reasons, and infections. And from what we know, all of these risk factors and injuries postnatally in extremely preterm babies lead to systemic episodes of hypoxia and inflammation, which are the main environmental risk factors for neurodevelopmental injury from what we know so far. And a lot of you already know this, but just like him, about 1 million babies are born extremely premature each year worldwide. And as the survival increases, we really, really need to pay way more attention, and it looks like we are trying to do that with this beautiful initiative here. So when we talk about extremely preterm babies and we look at the human gray matter, or like the cerebral cortex development, a lot of things are happening at the time when these babies are born, between 22, let's say, and 28 weeks of gestation. When you think about the neuronal development, a lot of the neural progenitor cells, which are going to form the neurons and the gray matter of the brain, are being born. A lot of them are migrating from the ventricular zone towards the pia to form the cortex, and the astrocytes are barely starting to form. And we know how important astrocytes are for the developing brain and for the function of the neurons. And then when we think about the white matter, we know that in the cortex, the myelination only starts after birth, and the cells that are going to produce the myelin are just barely starting to be made at the time of extremely preterm birth. So of course, this puts babies, extremely preterm babies, and for that matter, fetuses who are exposed to hypoxia for some congenital heart diseases, for example, or placental insufficiency, or other reasons, are also at risk for developmental injury long-term. And in the past, like 10 to 20 years, we've learned a lot about the risk factors associated with neurodevelopmental injury. We know more and more about the neurodevelopmental outcomes because of the beautiful clinical studies that we have. With advances in imaging, we know what the anatomic abnormalities are, both at the fetal stage, but also prenatally, postnatally. However, we still have a limited understanding of what is happening at the cellular level. And as a result, we have no biomarkers of injury or recovery for brain injury of prematurity, for example, and we have no effective or targeted clinical interventions to prevent this from happening, or at least make it better once it happens. And of course, this is primarily related to the fact that we don't have access to human, or we have very, very limited access to the human fetal brain or postnatal brain in the second part of pregnancy. And the other thing is that the fact that we don't have therapies or biomarkers for injury or recovery is not because of lack of trying. And we've learned a lot from rodent models, which are the most commonly used animal model to study prematurity and diseases associated with prematurity. However, for the brain, we think that we've reached a point where there are clear developmental differences, like interspecies differences, but also evolutionary differences in response to environmental risk factors like hypoxia or inflammation. And this is what's leading to the lack of, or like the minimal translatability of possible compounds or possible biomarkers or promising biomarkers from rodent models into humans, or like human therapies. For example, when we look at the developing brain, the organization of the inside-out organization from the ventricle to the PI is very similar in mouse and humans. However, what evolutionarily changed within like millions of years, of course, of evolution is that in humans and in primates, we have this subventricular zone that is highly expanded, that contains a lot of progenitor cells, neural progenitor cells, which continue to proliferate and divide up to 30 weeks in humans. And they contribute significantly to the expansion of the human brain just by having a larger pool of progenitors. So we think that these cells are primarily responsible for the expansion of the human brain in primates and in humans. And this is something that we cannot really study in rodent models because they lack that region of the developing brain. So in my laboratory and during my training, I was interested in developing these human cellular models for cerebral cortex development, particularly focused on the development of the cerebral cortex and the excitatory neurons and astrocytes in this region, but also on the focus on the development of inhibitory neurons, which migrate from the ganglionic eminences, which later on become basal ganglia, and then migrate in the second part of pregnancy to integrate with these excitatory cells here and to help the cortex function. So keeping in mind that essentially your models are wrong, but some are useful, we started using fibroblasts, and from fibroblasts, we reprogrammed those into human-induced pluripotent stem cells. And now we are able to grow region-specific cell types from various regions of the brain. And today I'm going to focus on the cortical organoids, which contain primarily excitatory neurons and astrocytes, and then on the ganglionic eminence-like organoids, which develop and form the interneurons, which migrate into the excitatory cortex. And not only that we can grow these separately, but we can put them together to study with live imaging how these cells migrate and how they integrate with each other to form a developing and like functioning cortex. Separately in my lab, we're focusing on oligodendrocytes and microglia because we know how important these cells are as well. And the goal is to form complex organoids to recapitulate as much as possible of the developing cortex and try to study these not only simple interactions, but also very complex interactions. Today, I'm going to show you two models and these two regions of the developing brain and how cells are affected. But before I do that, I'm going to explain briefly the model. So without going into details, practically we take these colonies of induced pluripotent stem cells, we put them in low attachment plates, they form these balls of cells, and then with specific growth factors, we tell them to become either excitatory brain cortex or inhibitory cortex. And one of the advantages is not only that we have human cells in a dish, but we also have lots and lots and lots of them, so we can grow thousands of them at the same time. And when we look inside, we see that they form these organized regions where with looking with specific markers, we can say that this is a ventricle-like area, this is the ventricular zone, this is the subventricular zone with those cells that we see highly expanded in the human brain, and then most superficially is the cerebral cortex with the neurons. When we do transcriptional profiling, we realize that after about 80 days in culture, our organoids, the excitatory ones, for example, transcriptionally, they resemble the 16 to 24 post-conceptional weeks fetal cortex, which was very important to me because I was interested in studying prematurity related problems. But as you can see, this can be applied at earlier stages to study different developmental stages in the fetal period. We have not only that we have neurons and progenitors, but we have astrocytes, and this is how they're supposed to look like. They're beautiful. And then they have synapses. And so the neurons are connected through synapses in complex networks. And using this model, this is a very simplified and fast version of the model, we studied the defects in the generation of cortical excitatory neurons following exposure to hypoxia. So after about 75 to 80 days, we expose them to hypoxia, and I can answer questions about how we chose these conditions. But practically, we showed that when we measured the oxygen level at baseline, we have partial pressure of oxygen of around 65 to 85 in the organoids, and then we dropped the level to around 25 to 30. in hypoxic conditions, and that's enough to induce hypoxia in our organoid models. And then when we did transcriptional analysis, we saw that the region that was the genes associated with cells that are in the subventricular zone, which is the zone that is highly expanded in primates and in humans, are the most sensitive to this exposure to hypoxia, which was very surprising, but also fascinating. So when we looked at these cells at the cellular level, not just transcriptional level, we found that indeed, when we expose organoids to hypoxia, we see a decreased number in these cells in organoids, and then this can be rescued with a compound called ISRIB, which inhibits the unfolded protein response pathway, which is a stress pathway that can kill cells under stressful and prolonged stressful conditions. This compound was discovered by Peter Walter at UCSF and is now in clinical trials, or like they're not in clinical trials, but they are trying to get it into clinical trials for neurodegenerative disorders because they have very, very good outcomes when they look at traumatic brain injury or trisomy 21, for example, and behavioral deficits associated with that. What we found, and was also very interesting, is that this decrease of cells was not because of cell death, but it was because the progenitor cells were prematurely exiting the cell cycle. So what happens in the end is that you have, by repetitive exposures to hypoxia or prolonged exposure to hypoxia, you have less progenitors to make neurons. And this could be one of the contributing mechanisms by which we know we see smaller and less complex cerebral cortex in babies and patients later on who are born premature. With help from the family planning research team here at Stanford, we're extremely fortunate to be able to work with them. We were able to validate all these findings in human fetal brain at around 20 to 24 weeks of gestation, and I will not show that data, but practically we show that using both of these models, we can see the same thing, which tells us that this could be very relevant for what actually happens in the developing human brain. And then last, I'm going to tell you about what happens to the migration of these inhibitory neurons and why am I interested in them. So we know that they represent about 20% of the cortical neurons in general, and they're very important for excitation inhibition. We know balance, and we know that when there are problems with their development, there is a clear association with neuropsychiatric disorders and epilepsy. So they're really, really desperately needed. We also know that they migrate in the second half of pregnancy. The peak migration is in the second half of pregnancy, which premature babies are completely missing in utero. And when we look at toddlers who are born preterm and have died for other causes, we know in histology that their numbers are dramatically reduced. And we know that preemies are at high risk for seizures and other neuropsychiatric disorders. So we exposed this, we generated organoids of two different regions. We put them together, as I showed you briefly, and we made the inhibitory neurons glow, practically tagging them with GFP, and we looked at their migration. And you can see here how they migrate after they transitioned into this excitatory part of the cortex. And when we expose them to hypoxia, what we found is that they just don't migrate anymore, which is really, really interesting. This was just one episode of hypoxia at a partial pressure of around 30 millimeters of mercury. And then the other thing that we found was this gene called Adrenal Medulline, which makes a protein called Adrenal Medulline, was very, very upregulated. When we looked at what this gene does and what this protein does, it's primarily secreted by the endothelium, and it's now being used in clinical trials for septic shock because it's very strong anti-inflammatory responses. But what we found when we added this adrenal medulline to the cells was this is control, this is hypoxia, and this is another control and hypoxia with adrenal medulline, and this completely rescues the phenotype that we see with the deficit of migration. And again, this is validation in human fetal tissue. You can see that there is a lot of these cells in the human fetal tissue, even at 22 weeks of gestation, and we are seeing exactly the same thing and exactly the same rescue. So these are two examples of the projects that we are doing in our lab, focusing on extreme prematurity and fetal brain development and injury, specifically focused on hypoxia. And I hope I've convinced you that that hypoxia, it's worth thinking about, because key developmental processes are affected and we desperately need effective therapeutics. And I also hope I convinced you that we can use human cellular models to try to tackle some of these questions, of course, with the goal of getting everything in vivo and hopefully in the future in clinical trials. And with that, I would like to thank my very young group of people. I have very, very talented people I'm very fortunate to work with. This is Haven. When he was 1 year old, he came to give us cookies. And this is his mom and his grandpa. So I'm still in touch with them. And I want to thank all of our collaborators and specifically the family planning research team, because this is a tremendous resource for us to try to understand what happens during the fetal life, not only with the brain, but also with other organs. And again, not only Taylor, Natalie, and Aisha, but also all the medical providers. You are all fantastic. So thank you for working with us. And thank you all again for inviting me.
Thank you. Thank you so much, Anca. That was fascinating and so thought-provoking. So maybe we can get everybody now together. And, this is meant to be really informal for the next couple of minutes. You don't, if you want to put a question in the chat, you can. If you want to just speak up, you can. You're unmuted. And we have several minutes. So I'll let other people ask. Otherwise, I have a few questions myself. But does anybody want to ask a question for Carl or Anca or David?
I have a question for Anca.
Go for it, David.
And maybe Carl too. So in my work, I've found HIF to be sort of developmentally regulated and really has some very different properties cell to cell, and even those cells across development is quite different. I'm wondering if, you talked a lot about hypoxia. Love to hear what you have to say. And obviously there's a lot of work in HIF in IBD. I don't know if there is any in NEC.
Yeah, I can start, Carl. So yeah, thank you for the question, David. I thought it was fascinating to see your data that it is in the lung developmentally regulated and it only starts getting expressed in later stages of development, because I think we talk a lot about this need for hypoxia during development. However, if you look at studies, we know that the cells are not hypoxic, and we keep talking about that. And I think what we're trying to study right now in the lab and to demonstrate more than study is that everything that we see, like the metabolism and everything else, everything that the cells do are HIF-1 independent in early stages of development. And the metabolism can be completely regulated towards glycolysis, which the cells early in development seem to prefer versus oxidative phosphorylation, even without HIF-1 alpha activation. However, when you have HIF-1 alpha activation, for example, there is a switch in metabolism towards glycolysis. So I'm really fascinated by that question. We're trying to prove that in our model, because I think a lot of the cells don't care about oxygen. Even in the presence of oxygen later, early on, they would not do anything with it.
Yeah, we always forget that red cell mass is so tremendous in the fetus that they may have a low PO2, but they're not hypoxic.
Exactly, and that's one of the questions that I get a lot. And one, like when I put in grants, it's like, yeah, but the fetals develops in less than 30, but then when you think that their hematocrit is like 80 and they have fetal hemoglobin and the cells are not hypoxic, clearly it's not the same thing. So the partial pressure of oxygen is not a direct sensor for oxygen within the cells. And I think we should all recognize that. Yeah.
Carl, I don't know
Carl, I'll let you take it on.
Yeah, I would just make a couple comments. So the thought that hypoxia is a significant contributor to necrotizing colitis, I think, has fallen out of disfavor. But to make this a smarter answer, what I would say is that we do know that why is it that 28 to 32 week corrected gestational age is the peak incidence of necrotizing colitis across every study that you could look at. My personal belief is, I think what you're speaking to a little bit is that developmentally there's cells susceptibility or vulnerability because of various pathways. And in the gut, in a mature animal or human, the stem cells actually are much more sensitive and much more prone to injury from various metabolites that are not buffered when you don't have a mature villus. So this villus crypt access really has to be well established, like in a term baby or an adult, to be less susceptible to handle all this metabolism, because they have very different routes of metabolizing, whether you're a crypt stem cell or associated cells or you're a crypt villi. And then of course in the premature baby, this morphology is not set up yet, and consequently the function is not set up yet. And we think that's what contributes to the predisposition, or you really only should say necrotizing colitis in a preterm baby, pneumostosis notwithstanding, which you can see in a variety of other ischemic events.
Thanks, Carl. Looks like Dr. Roncarolo has a question for you also, Carl.
Yes, hi, Carl. I really enjoy your talk, fascinating. Of course, as an immunologist, I cannot resist to ask you about T regulatory cells in the gut, because you have this cartoon on Th17 and Th1. But I personally think that, the most important cells in the gut are the cells that maintain the homeostasis and select between what is a pathogen and what is really a non-harmful antigen.
Yeah, so thank you for that question. I think that these cells were actually what's circulating. So what we find is that there's some programming that occurs in the gut, and then you can detect the differences in the cells that are outside the gut. And of course, I think the paradigm that you're setting up is, this is much less about, is this you're fresh out and this is an innate immune response from all the innate effector cells, whether it's a neutrophil or a macrophage. That would be your first encounter. They're not smart, they're not educated, and they're trying to reject or accept these different pathogens. I'm rather trying to speak to a physiologic setting where, we're lined by Enterobacteriaceae, as I said, a very pro-inflammatory, and yet you're not getting, most of us, or most babies actually don't have a significant problem from that. So it's really a peripheral effect around the Tregs that are there. I don't disagree with you around these first innate interactions as far as an immediate response before the immune system has been educated, being principally responsible in the gut itself. But there is some programming that will occur in the gut for education that will have effects systemically, which is, this is not my work, but we're trying to make the connection between how is the gut educating your predisposition or not to autoimmune disease later on in life.
No, I fully understand. And as I said, it's fascinating. But I think that's really what happens in the gut and in the draining lymph nodes determine, it's antigen specific. It's not polyclonal, it's derived. So it's not against self-peptide. It's against the peptide that the cells are exposed to. But we can discuss it later.
Thanks. Does anybody else have any other questions? David, I have just one question. The only on the prenatal side, our mainstay therapy for promoting lung development is betamethasone, right? It's been around for, God, since ligands and 60 years, and I was fascinated to see your diversity of all the cell types that are actually being identified now, and we're targeting essentially one cell, the type 2 pneumocyte development with our therapies. So any thoughts about, of all the cell types that you've identified, like any specific targets for prenatal therapy for lung development, not for malformations, but just promoting lung development in utero?
Well, I mean, we're looking really not necessarily for in utero lung development, which is a great question. We're trying to figure out targets to enable, to motivate postnatal lung development. But it'd be, because if they're in utero, I understand the issue, obviously, but you're going to have preterm infants. So we're really looking to find molecules that might play a role in allowing for us to do postnatal lung development. Relative to prenatal lung development, it's not really as if betamethasone is doing anything for lung formation. These kids are still born in the canalicular or some even in the saccular phase. And what we're really doing more than likely is just causing for excretion of surfactant from the type 2 cells, maybe some maturation, still not an altogether settled story. But one very interesting thing is the question of whether or not steroids exposure diminishes radial alveolar count. So that by giving steroids, whether or not in some ways you're compromising secondary septation, in favor of the surfactant protein maturation. And I know that neonatologists are always concerned about giving too many steroids because there may be the case, and there really is good animal data, that steroids do compromise the number of alveoli that you have. Now, we're not going to know whether that's a significant issue really until those babies are much older. But I think that our approach with steroids is, I think, quite primitive.
Thank you. Any other questions? If not, what we can do is take a 5-minute break and maybe regroup at 6:25. We might even be able to gain 5 minutes here and start our next section at 6:25 if that works for everyone.